So thank you for the introduction, and thank you to the organizer for inviting me and for organizing such a wonderful workshop, so many great talks. And I also would like to thank all the experimentalists in the room, because as a theoretician, your work enabled our research. And so I'd like to thank you one more time. Some of you are in the room, and I used the work of many of you. So as a theoretician, the fascinating thing of the genome, which perhaps I don't need to, it's a message that I don't need to hammer down with this audience. But the fascinating thing is also part that is slightly misconception, the fact that DNA is usually seen as a storage device. And that's probably the largest function, but DNA with all the proteins together form the genome, or at least the way I see it. And this ensemble of macromolecules, it works like a machine that processes information. And this is a field in which many physicists are getting more and more interested. This year, the Nobel Prize was to complex systems, to Giorgio Parisi. And complex systems are exactly this, are systems in which there are many agents that exchange contradictory information and contradictory signals. And somehow, they process this information. Now, for the genome, for all of these machines, the structure is connected to the function. In the case of the genome, there's some obvious connection between structure and functions that we've known for hundreds of years. For example, the change in structure along the cell cycle. More recently, we have come to know that the structure of the genome changes along cellular differentiation, and the structure of the genome changes along evolution. And again, I don't need to explain this too much, but most of what we know about this is due to these techniques of DNA proximity ligations. And to support what I just said, what this experiment have taught us in the past 10 or 15 years is that the structural ensembles of the genome in interface, they're not random, that they are specific to the cell line and not to the DNA itself, or at least not only to the DNA. And when you look at similar, when you look at the same DNA in different organisms that are evolutionarily related, often you see similar structures. So a few years ago, together with my mentors and colleagues at the Center for Theoretical Biological Physics, we set up on a journey to, write down a theory for chromosome folding. So most of what I'm talking today, it's about the chromosomal scale. We're interested in the shape of chromosomes, not in the shape of genes, which are too fine, too granular for these models, but in the shape of chromosomes. So the theory goes in 2 steps, which I'll illustrate sort of for historical purposes, but also because they help understand the more recent developments. But the theory goes in 2 steps. The first step converts ChIP-seq information, which is chemical information, into a sequence of chromatin structural types, which I'll explain what they are. And this is a machine learning step. And the second step is a physical simulations, molecular dynamic simulations like being introduced. And they convert this sequence of chromatin structural types in an ensemble of structures, and then with these ensembles of 3D structures, we can do all kinds of tests. So the first thing I developed was actually the physical model, which is called Microbe, from Michele's chromatin model or minimal chromatin model. Little I know that the reviewer disagreed that the model was not minimal enough for him. But in any case, so the overarching assumptions in the modeling of the chromosomes, they are all sort of like mediated from what we know about structural biology. So for protein, we know that there exists a sequence of amino acids, and this sequence of amino acids encodes the three-dimensional fold of the protein. So the ansatz of the minimal chromatin model is that there exists a sequence of chromatin structural types, and this sequence of chromatin structural types encode the fold of the chromosome. In this case, different DNA can fold in different structures, so the sequence of chromatin structural types must be something beyond just the DNA sequence, and we'll see that it's mostly related to epigenetic information. So with this sort of initial assumption, we went and we made two physical ansatz. The first physical ansatz is that there exists some kind of chromatin structural types. It's a block copolymer. If a wet two-segment of chromatin form a contact, the energy of that contact depends on the identity of those two, the chemical identity of those two pieces of chromatin. And this is, of course, inspired by the fact that if you look at the Heisey maps, you have compartment patterns, and these compartment patterns, you can label them. This is done with k-mean here. And these patterns, they align very well with the epigenetic information. For example, here you have this pattern here is B4, and aligns with H4K20 trimethylation, as you can see. So, and even more interestingly, when you have a change in the folding pattern, like you have here, that you go from dark light to light, dark, this is both cell lines is human DNA, so it's the same DNA. What you see is that associated with this change in structure, there's a change in methylation pattern. In this case, it's H3K36 trimethylation. Now, it's very easy to collect the structural information and verify that some epigenetic markers are more frequent in a compartment versus another. It's a little bit more difficult to invert this data and figure out what is the compartment given the epigenetic information. Because, for example, this compartmental change is H3K36, but this one is H4K20. So it's a little bit, it's not impossible. It's actually not difficult, but it's not trivial. So this was the inspiration of the block copolymer ansatz. And of course, this is always meant to be an effective description of the folding. So the actors are the proteins and the RNAs. We've been learning more and more. And those are responsible for the real interactions. And these are ideas that developed sort of somewhat independently in the field of condensates and in the field of genome architecture. Specifically, I would like to acknowledge these two papers that are the one that gave me the idea of phase separation, the paper from Barbieri on string and binder, and the paper from Daniel Jost and Cedric and Giacomo Cavalli on epigenetic markers. So this was the, and of course, we don't need to keep track of all these proteins to make a simulation. We can do what physicists do, which is doing a mean field approach. You take all these interactions, you lump them together in an effective free energy. And this is being done over and over again. For example, when you study the folding of a protein, you often don't take into account all the explicit water molecules. And keep in mind, the folding of a protein, it's really a property of the solvent, of the water. And here's the same. So you can do an implicit water protein, or you can do an implicit protein chromosome. So nothing is new in this sense. The second physical answer that we made is that there exist interactions that depend on the genomic distance. Now, at that time, when I made this assumption, extrusion was not known yet. Both groups were still working on their papers. So there's a couple of different sort of ideas that can explain why we made this choice. The first is that the decay of the probability with respect to the genomic distance is very specific in chromosomes and does not fit what is known about sort of more elementary polymers. So one way to think about this term that depends on the genomic distance is a term that can sort of enforce this power law scale. The second is the history of protein folding has told us that when there's a structure, you can often capture that structure with an energy term that depends on the translational invariant that depends on the distance. For example, in an alpha helix, an interaction between amino acid I and amino acid I plus 4 enforce the structure of the alpha helix. So whatever was the structure that I didn't know at that point, this could be a term that could discover that structure. And of course, again, the actors are, this potential is really representing the effect of the proteins. So we need to think about that. This interaction includes the effect of all the molecular motors that are acting along the polymer, and therefore they can know about the genomic distance between two loci. And then, some that were known, topoisomerases and polymerases, and then, the activity of SMC complexes was discovered with extrusion, and it made perfect sense. And again, this is a model for chromosomes, so we're not very much interested in the position of the CTCF. They're usually pretty close, in the scale of 100 kilobase. We're more interested in the activity of this model at chromosomal scale. So not very close to the diagonal like in this figure, but even very far away. So given these two answers, you come out with a Hamiltonian. This is effective free energy function. And you have polymer potential. And then you have this type-to-type interaction that includes interactions that act exclusively across space. And then you have interactions that depend on Cartesian distance and also genomic distance, which are the interaction along the polymer. As Bean has nicely introduced for me, you can take a Heisey map, and you can learn what are the parameters of your Hamiltonian from the Heisey map, just like it's been done in proteins for many, many years. So we learn about protein folding by looking at the crystal structures, and AlphaFold is just the last iteration of this process. But we learn energy functions from structures, and this is what we did in this case from Heisey maps. And what did we learn? Well, Heisey tells us that the B interactions are stronger. And this confirms what, this is a result from 2016, but this confirms what Leonid Bini was saying. So we have a case of convergence between different approaches and different groups, which is actually very interesting coming out of this meeting. Many groups were converging to the same answers, and that's a sign that the field is maturing. We're getting to good, we're doing good science. So the B interactions are stronger. And the second thing we learn is that this term, which at that time we called the ideal chromosome, because we didn't know what would come out of the data, but what came out of the data is that the energy term that depends on genomic distance is actually a lengthwise compaction term. And so short contexts are more favorable than long contexts in a specific functional way. And that's probably the effect mostly of these SNC complexes. Or there might be other models that we don't know yet. But this is a fact because it comes out of the Heisey maps. This is what the data tells us. So now we have this part. The second part, which is really the first part, is converting the epigenetic information into a single sequence of chromatin structural types, which is a problem that we solved with this machine learning method, which is, depending on your field, you can call it a spin glass. If you come from the physics, you can call it a half-field neural network. You can call it DCA. You can call it a recurring neural network. But in any way, it's a way to learn from the data once again. You can align the compartment annotation that comes from the Heisey maps with the set of experiments that you can get from the ENCODE database, for example. And at certain locus L, then you can read down this way, and you read the compartment annotation, and then you read a set of experiments. Then you have a database of those, and you can cook up your bioinformatics, and then you can take as an input these experiments and produce as an output the compartment annotation. And really, one of the most difficult things was coming out with the name of the method, which I'm particularly proud. So now we have full pipeline. You can go in a database. You can pick up ChIP-seq information, just chemistry, and take that ChIP-seq information, convert it into structural types with megabase, then use that input to molecular dynamic simulation with Micron, produce an ensemble of structures. And then you can check if you're doing well. And this is how you do in predicting the Heisey maps. They're almost indistinguishable at this scale. This is a 50 kilobase resolution. So here you have the simulated one. Here you have the experimental one. You can see the compartment patterns. The power scanning is also correct. And so this was the first test. Then we started looking into imaging because Heisey data, they're very different from imaging data in the sense that Heisey data, they're frequency data. They're not distance data. So frequencies and distances are really two different things. So with Heisey, you wouldn't know if the cell is this big or this big. It's just relative. So in this case, we were actually comparing with distances. This experiment was done originally in 2009 to show the properties of compartmentalization. And what they did is they put 3 probes in alternating compartments. And polymer physics would tell you that if you're farther along the polymer, you're farther along in 3D space. That's what a normal polymer does. And if you look what happened here, that's not happening in this chromosome, in this fish experiment. And you see that these two loci, L1 and L3, they're equally as far away than L1 and L2. So that's sort of like a mild violation of a normal polymer behavior. And in this other case, which the compartment, instead of being A, B, A, they're B, A, B, the violation is much stronger. So L1 and L4 are actually closer than L1 and L3. So there are 2 violations. One is a weak violation, one is a strong violation. And in both cases, the simulation was able to predict exactly that behavior. And mind, these are distances in micron that have been predicted straight from ChIP-seq. So distances in microns out of chemistry, which is very surprising and remarkable, I would say. So that was the first foray in the in imaging. That I have more later. So one of the other things that we immediately noticed in this paper, 2016, which actually Bing was an author as well, and was that the active chromatin tend to be positioned at the periphery of chromosomal territories. And we wrote it. We made a figure. We wrote it there. And that was true both if you looked at the active compartments or if you looked at RNA-seq. So you can take RNA-seq and map it on the structure and see where is the RNA that is actively described. And you can find that it's positioned at the periphery. And we've seen that the group Xiao Zhang has sort of verified that more recently, that the active chromatin tend to be at the interface between different chromosomes, which is it's fascinating. It's a fascinating notion with unknown repercussion at the moment. We also went forward and we see that when we do different cell lines, and there are compartment switches between different cell lines, what you see is really this chromatin that moves to the periphery sort of to confirm the same message. And this was done by Ryan Cheng in a more recent paper. So that's things that we learned from the structures early on. Then there was this question that has been bugging us for years, because people were asking us, like, but what if you have multiple cell populations? But this is an ensemble method. How do we know what really is the ensemble? Do I have multiple ensembles? Do I have one? Is it one structure? Is that one energy minimum? Is it flat? Is it fluid? Is it the soul? Is it a crystal? So we didn't know. But what we did in 2017 is that we made this figure to show what our model was telling us. We didn't know. So this figure was made to show 3 things. the importance of the proteins, which act to create this phase separation, the fact that the phase separation is microphase separation, not full phase separation, and the third thing that was meant to show was this dynamic character of microphase separation. So the fact that the microphase is the split and fuse, and so this is what we proposed in 2017. And then, thankfully, I mean, technology has moved on, and now we have DNA tracing, and we can go and actually check our structures with real ensembles of image 3D structures. This was done again by Ryan Cheng. And the bottom line is that so far it seems what we see in imaging is consistent with what we have in simulation. We don't really see a single energy minimum. We don't see multiple populations. The variability is well reproduced by the model, and here you see the radius of gyration. And somewhat disappointingly, you really cannot find, or at least we haven't found a good reaction parameter, but we couldn't find a way to cluster the different structures that are present in the ensemble. So what we find a continuum that goes from closed structures and open structures. The open structures are about some few percent of the ensemble. That's true both in experiments and simulations. And we see this sort of like, at least for this segment that was maybe 2 megabases, was imaged by Xiaohui Zhang. We see this sort of like dumbbell opening and closing, which perhaps is some similar idea to what Hibrensis showed us. But I don't know. But the bottom line is that so far, we seem to reproduce the variability fairly well. And I think this is another one of the notions that is being more and more established, that it's fairly fluid. There certainly is not a single minimum. The system, the method is predictive. We trained it once in 2015, and we trained on GM12H78, this one, and not this chromosome. We used chromosome 10. And then we saw that it's transferable to other cell lines. You can see that we can predict the structure of different human cell lines. In this case, chromosome 21 is fairly different. You can see the bottom is experiment, at the top is simulation. We can predict them fairly well. And in collaboration between the Center of Theoretical Biology of Physics and NIH ENCODE consortium, they're actually producing an atlas of structural ensembles for all the cell lines that are producing data, ChIP-seq data and Hi-seq data. And by this point, we have tens of cell lines, hundreds of millions of individual chromosomal structures. We had a lot of data for our purpose. We had the Xiaohui Zhang data. We wanted to compare. We built tools to reproduce these experimental assays in silico. So we decided to put everything, make it available to the community. So we created this Nucleon data bank, which is the work of Vinicius Cortesolo, who's sitting there mostly. And so, you can go and check this website, and you'll find all our ensembles, all the hundreds of millions of individual structures. You'll find all the tools to convert, to do the simulations. You find megabase, you find microbe, you find ways to do the in silico experiments. You find some amount of experimental data, and we're happy to host more when this is more produced. We put some data our own because it's our own interest, but if you have some data you want to publish, please send it to us, we'll put it on together with the rest. So that's a sort of, it's a project that was for us. It facilitated our work and hopefully will be a good asset for the community. So we did look into dynamics. So the model, as you've seen, is trained from Hi-seq. So it's really a model meant to reproduce thermodynamics and not dynamics. However, we do perform molecular dynamic simulations, and the molecular dynamic simulations are performed to reproduce the canonical ensemble. So it's just Langevin dynamics. we prescribe standard diffusion to the particles. And of course, the particle, they fill the energy function that we impose, which is the micron energy function. And then we can go and verify how do they move in that energy landscape, these particles. So what is known in terms of the sum of what is known, I mean, more and more is known by the day, and I'm actually looking forward to work on more of this dynamical data that are coming out now in microbiology. But what was known at this point in time was that the dynamics of DNA, or of chromatin in vivo, is subdiffusive with an exponent of about 0.3. And it's viscoelastic, as you can see from this sort of negative dip in the velocity-velocity autocorrelation function. And is correlated, as was discovered by Alexandra, who's sitting there. So what we did these experiments in silico on our trajectories. And we do fairly well in the sense that it is subdiffusive, in the micro model is subdiffusive, and the exponent is approximately correct. We do see the viscoelasticity, which is somewhat not impressive because it's a polymer, so that's sort of like a behavior of a polymer. And we do see the spatial coherence. So this is not to say that the model is perfect. This is to say that the model is reasonable in terms of dynamics. And now I'm going to get to the last part of the talk, which I want to highlight the latest work and mostly the effect of molecular models. This is the thing that Leonid Mirny touched on. Again, it's important to understand that the DNA extrusion and all other possibly unknown molecular models, they act all the time. So the important thing is not just the dots that we see where CTCF is, which are the position in which the molecular models are stopped, but molecular models are working all the time, everywhere. And this has important repercussions on the topology of chromosomes and on a couple of other things that I'll mention. So the first one is that these molecular models, they remove knots. Immediately on, I think Bean also mentioned something about this, but immediately we discovered that the micron chromosomes, they didn't have many knots. And that was a surprise because the high-C data doesn't know about the knots because it's a pair kind of data. It's just IJ, so you don't have topological information. But if you go and look, you can see that the micron chromosomes, they have a lot less knots with respect to a homopolymer, which is supposed to be knotted. And the longer the polymer, the more the knots, and DNA is very, very long. So there was this finding, which was very interesting, surprising. And that was sort of a reversal of paradigm, because for a long time, the paradigm was this one from Grossberg and co-worker of the fractal globule or the crumpled globule. And their description was, I take a polymer, I crumple it. Because of the crumpling, that doesn't have any knot. Then I don't let it chain cross, which means that I have very little topoisomerases. And because I don't allow the chain crossing, this polymer is stuck in a non-equilibrium conformation, and it remains without knots. If you were to let that polymer equilibrate, that would equilibrate to the equilibrium globule, which is knotted, because a polymer is supposed to be knotted. While here, there's a reversal, because we have an equilibrium state. We do allow the chain crossing. We do allow topoisomerases activity. We do allow the system to equilibrate. And by equilibrating that polymer, actively remove the knots because of that ideal chromosome term, which is a lengthwise compaction, which is connected to the activity of sensory complexes. So, and the funny thing is that already in 1988, there wasn't much wrong in the structures that they were obtaining from the fractal globule. So structurally, the fractal globule, it's pretty good. It's the process that we now think is not It's not a non-equilibrium process, but it's more of an equilibration process. Now, whether we do reach the full equilibrium, that's a different story. But here, again, the key here is that you allow chain crossing. And the key here is that you don't allow chain crossing. You don't allow chain crossing to keep it out of equilibrium, and you allow chain crossing to let it equilibrate and remove the knots. And that also connects to what Job Decker was saying today, which was very interesting. Anyway, so this was early on. More recently, we had another work about the effect of molecular models on the establishment of chromosomal territories. So mammalian architecture has chromosomal territories. This is a Heisey map that probably you all see. This is a famous picture from the Kramer and Kramer. The chromosomal territories, they show up as this square along the diagonal in the whole genome Heisey map, and they show up as these globules when you do imaging in the Kramer and Kramer figure. Now, what happens to other organisms? So this is a big paper, big collaboration, lots of people, and mostly Olga Dutchenko from the Eretz Lieberman Lab, and Claire Horenkamp from the Netherlands, from Benjamin Rowland group, and ourselves for the theory part. So what they did is this. We took 24 species, spanning about 100 million years of evolution, and we did Heisey, and we did the genomic assembly for these species. And these are the Heisey maps. There's a lot of variability. And this is a moment in which we stopped, and we scratched our head, and we thought about, like, how do we reconcile the fact that there's vastly different numbers of chromosomes, they're vastly different size, these chromosomes. How do we compare these maps? And then what we did is sort of like to renormalize the length of these chromosomes and the number of these chromosomes. So we just average all the chromosomes to obtain a single intrachromosomal map and a single interchromosomal map. And then you can go look, and now there's a lot more order, even though there still is some variability. But what you can't see in this is that these 24 species, there's only 4 features that recapitulate the genome architecture of these 4 species. And these 4 features are the existence of chromosomal territories, which shows up with the presence of the square along the diagonal, the clustering of centromeres or telomeres, which shows up as these strong dots in the intra or interchromosomal maps, and the presence of an alignment between centromeres and telomeres, sort of a fallback of the chromosome, which shows up as this sort of secondary diagonal in the maps. Even more interestingly, these 4 features are not uncorrelated. Typically, you have either the territories or a combination of these 3. Once you have one of these 3 features, then you're very likely to get the others. And this is sort of the probability of those, which is done, creating some order parameters and threshold in them. But it's fairly evident even from, it's fairly evident even from this map. See, here you have, for example, the secondary diagonal, and both. Here you have all these 3 features. Here you have this, and it's only the territories. So it was fairly evident, and then it can be quantified. Another very interesting thing that we noticed is that when you go look in the genomes of these organisms, and you look for what proteins are present, you find out that many of the organisms without chromosomal territories, they happen to be missing, to be lacking one or more components of condensin 2. So you can do that just by blasting their genome and see if you find any reasonable hit for that protein. So this is really cool. And this is when I woke up. I was like, I know why that happens. And this is the micromodel, right? So in the micromodel, you have the polymer. That's fine. That's just connectivity. And then you have the type-to-type interaction, which I said this is epigenetics. And then you have the ideal chromosome, which I said this should be probably motor activity. So if I lose one of the SMC motors, one of the extruders, then I should have that this term which is the lengthwise compaction, goes down. And Sumidaba, who's sitting there, did the simulations and spearheaded this project. Lo and behold, when you take a normal chromosome, wild type, with territories, then you lower the lengthwise compaction, you lose the territories, you gain the clustering of the centromeres and telomeres, which are heterochromatic region. They always wanted to phase separate. They just couldn't phase separate because of the motor activity. The moment you remove the motor activity, then this phase separation may happen. So this was done in silico. It's essentially a condensing 2 knockout in silico. And then our friends in the Netherlands, they did the knockout in vivo in human cells. You go from territories to clustering. And this figure is to show the fact that the clustering is the phenotype of flies. And they thought that the fly was a good rhetorical argument. Anyway, so this concludes my talk. I think we showed that at this level, chromosomal scale, there's a theory, and the theory kind of makes sense, and it represents many of the things that other groups have found by doing more detailed modeling of the activity of these proteins. And the theory also connects, again, perturbation experiments and connects molecules to their effects, and to their effects at very large scale. And interestingly, along evolution, this sort of like loss of function of condensing 2 is a thing that happened multiple times independently. So we don't have a single branch of the phylogenetic tree that developed that feature and kept it forever, but kind of like switched back and forth, which I'm not super expert in evolution, but I find it fascinating. So I'd like to thank all my collaborators, my group at Northeastern, all my collaborators, CDVP, Jose Arnicic, Peter Wallin, Ceres Lieberman-Aiden, Ting Wu here in Boston, Olga, Vinicius, Sumidaba, Antonio there that's leading the software engineering development, Benjamin and Claire, which were the main motor of the Condensing 2 project. I like to advertise that we're basically always hiring. There's position in my group, there's position at CTPB in Boston, there's position at CTPB in Houston. Here in Boston, we are in beautiful Back Bay here at the 13th floor, and I'd like to take questions. Thank you.
Hi, my name is Atta. I'm a grad student in Dave Thirumalai's group. A very interesting talk. I believe you mentioned that in your model is kind of like an Ising model with interaction parameters for corresponding to each pair of loci. You showed that the model already shows that the B interactions are stronger than the A interactions. So I was wondering, have you tried coarsening the interaction parameters, like making neighboring loci force them to have equal interaction parameters to check, if you reduce the parameter space, how does it influence the conclusions?
So this is a very good point, and we haven't done, so I think you're hinting to using some kind of renormalization approach.
Yeah.
We haven't done specifically that, but we have variety of model. We did coarse-grained. We did coarse-grained to larger size in terms of beads, more chromatin in a single bead. We did coarse-grained in terms of number of chromatin types. Everything checks. But the reality is that it's not that difficult to get these things to work.
Okay.
So to be fully honest.
Okay, thanks.
I have a question. So for your models, you're taking in sequence data from biochemistry, pulling down different marks, and then building a model, a physical model. You can get to the actual map that you would see with distances for microscopy.
Yes.
Is it possible, what's the resolution? What's your limit on resolution, I guess? And for some of the techniques where you can get more fine-scale mapping, like a micro-C, can you build models that you think would be reasonable?
So all this work, it's about the same scale. The beads are about 50 kilobase to 100 kilobases. I purposely left out all the newer development in terms of different scales. So it's about almost 10 years of being working in this field. So at this point, we have models at all scales. So we have a model that's probably going to be published soon, which is nucleotide resolution. We have a model that is nucleosome resolution. And we're working to get them all to work at the level of qualitative level of quality, quantitative level of agreement with the experiments that I think it's important to convince this audience that there's some good things in what we're doing. So they're not yet out, but we're going down. So in terms of, so we can do whole chromosomes. I mean, the center is working actively in keeping this level of resolution, but scaling up to whole nuclei. And in my lab specifically, we're going down all to the level of genes or nucleosomes. And they're all doing okay at the moment, but soon they'll be just right.
Excellent.
Great talk. In one of your last summary slides, comparing the equilibrium global, chrome to global, and micron global. So in the micron global, I saw that you mentioned that it's topoisomerase activity should allow the chain crossing the system. So is that right? You also implemented that kind of a chain crossing in your simulation?
Yes. So they implemented the chain crossing. It's crucial for two reasons. Well, two reasons, which eventually it's just one. We think it's correct because topoisomerase exists, so you should have chain crossing, and that represents what the biology is. But also, it's good for your simulations because you converge to the equilibrium that you want to converge. Because if you don't allow chain crossing, you don't let it equilibrate. So your simulation would take a lot more time if you wouldn't allow chain crossing. So yes, all the simulation, they allow chain crossing. We don't allow chain crossing in the nucleotide model for the moment because of other reasons. Because the timescales are different, and when you want to study the motion of nucleosomes and supercutting, it's maybe topoisomerase will come later on. But yes, all this has chain crossing.
But isn't your a homopolymer backbone based on a self-avoiding chain?
Yes, with a finite energy penalty.
Oh, I see. Finite energy penalty. Okay.
Which meaning that occasionally would allow chain crossing.
What's the rough estimate?
4 kT.
Any more questions? All right, let's thank our speaker then. We'll take a 10-minute break.